Hey guys, so welcome back to this session today. So this video is going to be all about asexual reproduction. So this is part 1 and so let's first study about what is reproduction. So reproduction is the process of giving birth to the new individuals of the same kind. So please highlight with the statement of the same kind. So I'm coming to say that producing new individual of the same species. Okay, a cow cannot give birth to a dog. Alright, a dog cannot give birth to a cat. So reproduction is the process of giving birth to individuals of the same kind, okay, of the same species. Alright, but there might be variations which we will discuss later. Okay, if the organisms they are not reproducing, their population might decrease and that particular species they might even become extinct. Alright, so reproduction is not a very required biological process. Like you have so many other biological process like excretion, digestion, respiration. So these are all very much required for the healthy living of a species. Okay, if you're not doing respiration, then you might die. But reproduction is not like that. If you're not doing reproduction, then it is a problem. No. Okay, so it is not a very much required biological process, but yeah, it is important because it will help in the continuation of species and for the survival of the population of that particular species. Okay, so first in this reproduction the organism they will create carbon copies of themselves. So DNA is the deoxyribonucleic acid which is present on the chromosomes in the nucleus. So that will be helpful or that will be responsible for the transfer of genetics. So another important feature in DNA copying will be variations. So variations are like little pattern difference. Okay, so while the reproduction occurs, there will be a little pattern difference in the DNA copying. So that is what we refer to as variations. So these variations are very well essential for an individual to survive. So there may be many changes, like there will be changes in the temperature and any environmental changes. Okay, so in such cases the individual can fight against it. Okay, so this kind of variation can help in the evolution of new species as well. Talking about the difference of asexual and sexual reproduction, first asexual will involve uniparental. Okay, so only one parent will be involved, but in sexual reproduction two parents are involved. In asexual reproduction there's no fertilization, like there's no gamete formation, but in sexual reproduction yeah, gamete formation occurs and fusion is there, fertilization is there. So and in asexual reproduction the progeny is the offspring. So the offsprings are very identical to the parent. So there might be no evolution at all. But in sexual reproduction, yeah, there's no particular identical at all, but they are little bit similar, but they're not identical. So evolution of species can be seen. Another thing is asexual reproduction is faster but sexual is slower. So that's all about the difference between asexual and sexual reproduction. So now let us study about different types. First one is binary fission in amoeba. As you can see how amoeba undergoes fission. So binary fission is simply the division of cell nucleus into two. Bi means two, right? So the division of cell nucleus into two parts is what we refer to as binary fission. So first there is parent amoeba and then there is elongation of nucleus, division of nucleus, and then finally the cytoplasm itself it will divide and then two daughter cells will be formed. So now for multiple fission, which we see in the malarial parasite called Plasmodium, so in which many daughter cells are produced. So first of all, the most important thing is that in multiple fission cyst formation will take place. So cyst is like a thick outer layer. Okay, a thick out layer will be formed and inside that one cell will divide into many daughter cells. So multiple fission, right? That's what. Okay, so many daughter cells are produced by multiple fission and finally when favorable conditions are happening they are released. Okay, so the major difference between binary and multiple fission, let us see now. So in binary fission, okay, so it will form only two daughter cell, but in multiple fission many daughter cells are formed. So in binary fission, nucleus of the parent will divide only once, but in multiple fission it can divide many times. Okay, and binary fission, it's a very very important point. So this can only occur during favorable conditions, but multiple fission usually takes place under unfavorable conditions. Okay, so unfavorable conditions, it's a mistake in the picture. So in binary fission you can take example as amoeba, which is a very great example. Multiple fission you have plasmodium. Okay, so now for fragmentation is simply the once the organism will attain maturation, the body parts of the organism will divide into fragment, and each fragment will have the ability to grow into individual organism, and mainly Spirogyra. So it undergoes fragmentation. So let us understand how it undergoes now. So as you can see in the picture, so the body of the Spirogyra will divide into fragment, and that each fragment will have the ability to grow into individual Spirogyra organism. Okay, so now we will talk about regeneration. So guys, first you have to understand that regeneration is not the same as reproduction, because many organisms, they would not normally depend upon being cut to be able to reproduce, because regeneration is all about planaria. So if you cut any part of the planaria accidentally, the each part of the planaria can grow into individual planaria organism. So you could have already seen lizard. So your parents used to tell you the story, right? If you cut the tail of the lizard, it will grow, the tail will grow again. Okay, so that will how it will happen in planaria. So that's how it will happen, but there's a huge difference. Okay, so in lizard, that tail won't grow. Okay, that body will have a outgrowth of a tail, it will grow the tail, but that tail will remain lost. Okay, but in planaria, if you cut the organism into two parts, the head will grow into a full body, the tail will also grow into a full body. All right, so planaria or other organisms, other type of organisms, they will also undergo this type of reproduction. Definitely all of us will have this confusion. What is the difference between fragmentation and regeneration? They look similar to me. Okay, so fragmentation will occur in multicellular organism with a simple body organization, but regeneration will occur in a multicellular organism with complex body. Mainly you have to understand that in fragmentation, it itself will develop into fragment, and each fragment will be growing into new organism. But in planaria, we have to cut it, and only the cut parts will be able to reproduce. All right, so an organism, it cannot simply depend upon being cut to be able to reproduce. So that is a major difference. So in fragmentation, an organism will break into individual pieces. In regeneration, if it breaks into pieces, then it might be developed. So now about hydra. Hydra undergoes reproduction by budding. Okay, as you can see on the picture, first there will be a parent hydra, and there will be a small outgrowth called bud. Okay, the bud will develop, and after maturation, it will detach from the parent, and it will become a new hydra organism. So budding is basically the process of developing an outgrowth in an organism. So major example for budding would be hydra and also yeast. Okay, so as you can see here, this is the budding in yeast. It will form chain of buds, and it will develop into an organism. So now let us talk about vegetative propagation. So these are the common types you can see in it: cutting, layering, grafting, etc. So we do not have we do not have division. So cutting, layering, grafting, and tissue culture, let us look into that. So first about grafting. So grafting is basically cutting two parts of the plant, and we are joining it, and we are wrapping it together. Okay, so the two plants can be chosen, like you can mix two characteristics of different plants. For example, you took one plant which is of disease free condition, and the other plant which is a high yielding variety. So the new plant which so the result will be both high yielding variety as well as it will be a disease free plant. So that's how you can join two different characteristics into one plant. Okay, so this is a very good technique. The bottom rooted plant is what we call as stalk, and the top plant is what we call as scion. So scion and stalk are joined together, and they're wrapped together to form a new plant. So now let us understand about cutting. So cutting, rose will undergo cutting generally, stem cutting, sugar cane. Okay, so cutting is where one part is it's been cut, and it is put in the soil to be regenerating again. So now for layering. Layering is basically, taking one bottom branch of the plant and we are burying it in the soil. So you should not bury the plant entirely, but the growing tip should be above and the branch should be, covered with some little bit of soil at least. Okay. So this is called layering, and that part will be growing into new, plant. Okay. Wow. So next process will be tissue culture. So tissue culture is basically undergone by ornamental plants. Okay. So where you are separating the growing tip. Okay, growing tip, or you will separate the cell. So these cells you can place it in an artificial medium and you can add some kind of hormones for growth, like coconut milk, agar agar. So these are some kind of growth hormones. So you can use it and you can grow them into mature plants. So using tissue culture you can grow many plants from disease free condition. So this technique is for ornamental plants. So that's it, we've come to the end of part 1 video. I hope the video is helpful. Thank you for watching. Bye bye. So let's meet you guys soon on part 2 video.